Hi everyone. Today we will be finding a easy way to learn the biotechnology lesson so that it becomes easy for you to answer the questions both in board exam as well as in the NEET exam. So first let us go sentence by sentence from the textbook. Okay, so biotechnology normally what it deals with using live organisms or enzymes from organisms to to produce products and processes useful to humans. So it has a very simple meaning. We are going to use microbes or the enzymes from the microbes or any other organisms to have products, to make useful products. That's the basic idea behind this. Say for example, we are making curd. Curd or yogurt is made from the milk by mixing the probiotic culture which is already there with us. So that is a simple biotechnological process. The way we are making idli batter, that is again a microbial process which is a simpler form of biotechnology. So like that we have made pizza dough and so many other things. The dough we are making for pizza, fermented idli batter or dosa batter or any process we do in a very simpler form to get the fermented product comes under biotechnology. But this is a traditional method of using microbes in a very direct form. Instead, we want to work with the molecular genetics, that is the DNA of the microbe, then it comes to the modern biotechnology. That is what it has been explained here. If you are using microbes or any other organisms directly to get the benefits or the products from them, that is a traditional biotechnology. If you are manipulating genetic material of the microbes and deriving the products from them, then it is referred as modern biotechnology. So the many of the processes we are using nowadays, like making test tube babies, synthesizing new gene, developing DNA vaccine, all these are the part of modern biotechnological process. The European Federation of Biotechnology has given a definition. The definition is given here. The integration of natural science and organism cells, parts thereof and molecular analogues for products and services. We are going to integrate natural science and natural organisms, their cells and the parts of the cells and their molecular analogues. Analog means which is resembling something, resembling the molecules which is already existing in the cell. So the molecular analogues also we are going to use it. Why we are going to use this? To derive the products and services, the benefits we are going to get from them. So this is the basic idea behind this. This is a very simple paragraph which implies the difference between the traditional biotechnological process and modern biotechnological process. That means in our everyday life also we are using biotechnological process which doesn't involve much of the equipments and all, isn't it? So hope you understood this point. Let us move on to the next one. What is the principle of biotechnology? There are two main principles behind biotechnology. That is the modern biotechnology only we are going to learn hereafter as biotechnological process. So what is in the modern biotechnological process basically needed? The first thing, let us understand this idea first. First thing, you need to take a DNA from a desired plant or animal, whatever it is. For example, let us consider you have two mango trees. One mango tree is giving a nice juicy fruit, but the fruit size is very small. You have another mango tree which is giving a bigger fruit, but it is not nice, sweet and tasty and juicy. So what do you want? You want a bigger mango which is nice and juicy, sweet and bigger in size. So you want to incorporate both the qualities together in a plant. So traditionally what will be the method we will be doing? We will be doing hybrid. So you will cross two plants and wait for the seeds to be produced, and once the seeds are produced, you will identify which plant will give the better result, which seed is going to give the better result, or you will use plant tissue culture or any other such method. That is also biotechnological process only, but that comes under plant breeding techniques. But here what we can do in another way to have a quick result, I have to identify the segment of the DNA which is responsible for juicy, sweet mango. At the same time, I have to identify the gene responsible for the fruit being bigger in size. So I have to now take out both the genes, incorporate them so that I can get a new combination where the fruit is going to be bigger as well as tastier. Okay, so this is the basic. So I have to play with DNA now. So for playing with DNA, I need to use the genetic engineering tools basically. So that is what is being told here. We basically need two core techniques for this. One is genetic engineering, where actually you are going to play with the DNA and change the DNA sequence basically. And then maintenance of sterile contamination free condition, microbial contamination free condition to get the desired product. Okay, let us take another example to understand this very clearly. Say you are using, we are using insulin nowadays to treat diabetes, actually not to treat, to combat with the problem of diabetes. So here what we can do is, we cannot take human insulin from any other human and inject into other person, which is highly impossible. Or they have tried various method of collecting insulin from other animals like pigs and all, which is also not very suitable for humans. Then they started making the recombinant DNA for insulin gene, and from then they started producing insulin. So basically how this was done, insulin RNA was taken out or insulin gene was taken out, then it was introduced into the microbe. So insulin gene from a human was identified and isolated. So for this, the big human gene, so say for example this is the human gene, very long gene, this is the insulin. So this part alone to be cut and isolated. Isolated means it should be separated and then purified. Then this gene must be introduced into any microbe. Now we have to identify the microbe which is having the gene and take the microbe which is having the desired gene, grow that in a larger scale so that as the microbe grows, it produces human insulin. So there are two steps in that very clearly. You are playing with the DNA, that is one step. Cutting the human DNA, getting insulin gene, introducing this gene into the host organism, that is a bacteria. That is the first step which comes under genetic engineering. The second step is, once you introduced it, identify the microbe which is having this gene and then grow it in the larger scale without being contaminated by other microbes so that you get the better product. These are the two core techniques or two main principles of genetic engineering. So with this I will conclude this video. Kindly take a time to go through this video. Thank you.